The most common and regular of the protein domains are protein domains that consist of both alpha helices and beta strands, and these are known as alpha-beta domains. Following along a peptide sequence that is part of an alpha-beta domain, what you would find is that it's alternating between a beta strand that's connected to an alpha helix, connected to a beta strand, alpha helix, so on and so forth. They repeat over and over again. Actually, it's not quite that way, because in between the beta strand and alpha helix is a loop segment, and a loop segment is a conformationally unstructured peptide segment. Here you can see a loop segment in the structure down here, which is relatively unstructured. That is, it doesn't have any kind of regular conformation. And it joins the end of the beta strand, the C-terminus, and joins up the alpha helix. There's two main packing domains. On the left, we see one of them, that's the alphabeta barrel. In the alphabeta barrel, what we have is a beta sheet that forms actually a closed loop, so this sheet comes around all the way to form the cylinder kind of object that's the interior of the barrel, and parked on the outside of that beta sheet is a set of alpha helices that more or less corral it and form an aqueous that's in contact with the aqueous phase. The topology diagram at the bottom shows the connectivity, and you could start, and I would encourage you to do this, to go ahead and work through this on your own. So starting with the N-terminus, that would be down here, we'll follow this through, first encountering strand number 1, then there'll be a loop segment, and we'll get into the first alpha helix. And so that's traveling along like that, and then there'll be a loop segment into strand number 3, strand number 2. Strand number 2 comes up again, and so you can keep going. And I would encourage you to go around and see how this alternates between the beta strand loop, alpha helix loop, beta strand loop, and so on and so forth. The second domain is really a sandwich kind of structure. It's called the alpha-beta open twisted sheet. As you can guess, the sheet itself is kind of twisted. On the top of this are alpha helices, and on the bottom of this sandwich is an alpha helix. And then there's a beta sheet that sits in the middle of this, and the beta sheet has a structure that we'll look at in a second. It's kind of hard to draw it, but it has this sort of twist to it. If we look at the three-dimensional view over here, you can start with the structure at the N-terminus and travel along strand 1, and then you get to a loop region, an alpha helix, strand 2, loop region, alpha helix, and then strand 3 ends up with going into a loop region that finally connects to strand 4. If we look at the connectivity diagram or the topology diagram, we can see that the first sheet is traveling away from the center toward the outer edge, and then it joins back up into strand number 4. So as the diagram shows, there's an adjacency between 1 and 4, just like you see here in the actual three-dimensional structure. Strand 4 is adjacent to strand 1, and that's represented in this topology diagram. It turns out that right in between strand 1 and strand 4 is the active site. So that's easy to recognize because, as I was saying, the sheets move from the center toward the edges, but notice how they splay out in opposite ways, and this is actually right where the enzyme active site is located. I would encourage you to go through and try to see the connection between this three-dimensional structure and the topology diagram, trying to pay attention to both the connectivity of the different secondary structures and also the adjacency of those structures. Let's take a little bit closer look at the two different packing motifs. The alpha-beta barrel consists of parallel beta strands, and so that's because they all enter into the barrel at the bottom side after looping around and going through an alpha helix and another loop. The outside of that barrel is all packed against alpha helices, and so you might not be too surprised to find that on the interior this is all hydrophobic amino acids, and they basically are going to make the structure very stable in water, where the alpha helices will have polar segments on their outer edges that will make this water soluble. And then finally we can see that the enzyme active site is located at the top of the barrel, the C-terminus of the barrel, and that's where we'll find, usually with loop segments, providing some important enzyme active site side chains to carry out the enzyme functionality. the alpha-beta open twisted sheet has alpha helices on both sides, and so it's like the sandwich structure that I was explaining. Not too surprising. There's going to be a very large amount of hydrophobic amino acid side chains in the interior to make this amphiphilic kind of structure, where again there will be polar side chains on the alpha helices. And at the C-terminus of these parallel strands, in that region, that crevice where the two strands splay apart and go in opposite directions, that's where we'll find the enzyme active site. Here's an example of a structure that is that forms the alpha-beta barrel domain, and right now you're pretty much looking down the alpha or the beta barrel, and then you can see that it's surrounded on the outside by these alpha side chains. Over here, let's take a close look at the strands that form just the beta barrel part of the interior core, and if we see that it's filled with side chains that will both pack the interior space and then pack up against the alpha helices, we can go ahead and do a space filling model of those, and the space filling model shows just how dense that interior core is. All of the white segments are side chains that are hydrophobic, the red are side chains that are hydrophilic, and so we can see that predominantly, as we look down this barrel, it's made up of a pretty densely packed hydrophobic core. And now over on this side we can see that the alpha helices pack around the core and form basically what will be an amphiphilic kind of structure. And the top, right at the top of that, where this segment is shown, that's the enzyme active site. This is the spinach glycolate oxidase enzyme. It's our prototype alpha-beta barrel. Now let's look at adenylate kinase. Adenylate kinase forms what's known as the alpha-beta open twisted sheet, and we're going to try to see how that sheet is actually twisted. Here, try to get a picture of this sandwich kind of structure. There you can see alpha helices on the top, above, and then this alpha helix down here, you can see that it's buried beneath the beta strand, and so there's this sandwich kind of layering effect. If we look over here at the next picture, we can see that the sheet itself is rather twisted. So there's the sheet all by itself, and if I stop the movie right there, you can see that the sheet definitely has an awful lot of twist to it. It's by no means a simple planar kind of object. So we can also look to try to find out where the hydrophobic amino acid segments are, and if we look at the sheet and color the amino acids that are hydrophobic white and the polar amino acids red, we see that by and large the sheet is made up of hydrophobic amino acids, and so that's going to make a lot of sense when we consider the structure that is going to be basically the sandwich kind of structure. The interior is going to be filled with hydrophobic segments, and then we'll have the amino acids that are going to be polar on top, and they will end up making contact with water to make this amphiphilic kind of structure. There you can see how much of the amino acid hydrophobicity, the large hydrophobicity of the amino acids. This is an enzyme that carries out a disproportionation reaction. It takes AMP plus ATP, so monophosphate triphosphate, and ends up making two molecules of a diphosphate. And so we'll be looking at the enzyme active site, this structure here, which is a bis adenosine pentaphosphate that is bound to the enzyme active site. We expect to find it at the C-terminus up here, where the two strands of the sheet are going to be splaying apart. Let's see if we can find that. So here's the structure, and you can see this sheet right now, and this is the enzyme active site, where this inhibitor is bound to. And right at the C-terminus, so we can see the arrow points to the C-terminus of the beta strands, and right in that region is where this active site is going to be found. So there's a picture, there's a couple of pictures that you can see that. There you can see it with space filling, and it's going to form a crevice packed in there fairly tightly.